Friends, it is a profound privilege to speak today in memory of my dear brother, friend, and colleague, Ilya Shmulevich. For over 20 years, I had the honor of knowing Ilya and working closely with Ilya. Reflecting on his impact, particularly in bridging cancer genomics and systems science, is both emotional and uplifting. I owe my deep gratitude to my friend Ed Dougherty for introducing Ilya into my life in the year 2000. Ed, a pioneer bioinformatics scientist and engineer professor at Texas A&M, was working with me on cancer genomics at MD Anderson Cancer Center. It was a magical time as our program was expanding. Ed told me, Ilya is one of the most brilliant scientists you will ever meet. You will never regret hiring him. When Ed speaks, you listen. We quickly processed the paperwork to bring Ilya to MD Anderson from Finland as a research scientist. Not long afterwards, we promoted him to tenure-track assistant professor. Thank you, Ed. Many of you may remember the early days of genomic profiling, primitive yet exhilarating. The first microarrays we used were clone-tech membrane arrays using p32 labeled cDNAs for hybridization. Later, we began printing our own microarrays on glass slides using Cy3 and Cy5 labeled cDNA for hybridization. We also handmade a number of tissue microarrays. In those early days, Ilya and I spent many hours working on microarray quality control and developing methods to analyze omics data. We co-authored and co-edited a couple of books, which was a lot of fun. My group was the first to profile gene expression in primary human glioma tissues using the first generation of microarrays, analyzing over 588 cancer-related genes. This work led to the discovery of IGF-BP2 as a consistently overexpressed gene in glioblastoma. Ilya and I often discussed glioma systems biology. We were keen to understand how transcripts were interconnected in gliomas and how to model this network. We set clear goals for our models. Our models should, 1, incorporate rule-based dependencies between genes, and this may represent key biological information. 2, enable systematic study of global network dynamics, particularly how individual genes affect long-run network behavior. 3, address uncertainty in our models, given small sample size and noisy measurement in most of real-world studies. 4, quantify the relative influence and sensitivity of genes in their interactions, allowing us to focus on key genes or group of genes. These efforts eventually led to the development of probabilistic Boolean network (PBN) model. which resulted in several highly cited publications. With this highly innovative work, Ilya submitted an R21, and it was funded on the first try. Then he submitted an R01, again funded on the first try. The only grant he didn't get was an NSF application, but even the reviewers noted that it was elegantly written like a textbook. I want to thank Stu Kaufman, whose brilliant work was a major inspiration for the development of the PBN model. Ilya and I brought Stu to MD Anderson for a visit, and we immediately fell in love with his ideas and personality. Thank you, Stu, for your brilliant minds and your friendship all those years. I have to admit that in the early days of our partnership in systems biology research, I was a bit nervous about mathematical modeling. While Ilya had many questions about molecular and cancer biology. We shared countless conversations and moments of laughter in our section of the hallway. I recall asking Ilya one day, how do I know if the genes relationships in the network are correct? He replied, that's your job. So we agreed that key relationship reviewed in the PBN models must be validated experimentally, and the network built from our model should provide valuable theoretical guidance for future experiments. As I showed earlier, our microarray experiments identified IGF-BP2 as being important in glioblastoma. Over the years, through the work of 3 outstanding graduate students, we've shown that it plays a key role in glioma invasiveness. IGF-BP2 binds to integrin alpha-5 through its RGD domain, and IGF-BP2 promotes glioma development and progression in the glia-specific transgenic mouse model. These studies led us to conclude that it acts as an oncogene, despite the absence of mutations. This also illustrates that cancer can be initiated without gene mutations, which is a topic for another discussion. I believe Sui Huang has a lot to say about this topic as well. Naturally, the first subnetwork Ilya and I wanted to study was centered on IGF-BP2. So let's look at the IGF-BP2 subnetwork based on PBN modeling using gene profiling data from 24 glioma patients. We immediately noticed a connection between NF-kappa B and IGF-BP2, which made sense because we had found an NF-kappa B binding site on the IGF-BP2 promoter. A literature search revealed a paper showing that NF-kappa B activated IGF-BP2 in lung epithelial cells. Furthermore, my student at the time, Hua Wang, showed that IGF-BP2 overexpression increased NF-kappa B activity in reporter gene and DNA binding gel shift assays. In our IGF-BP2 network, it was linked to NF-kappa B through integrin-linked kinase, ILK, and TNFR2. This result excited us, Our real-time PCR data confirmed that IGF-BP2 enhanced ILK expression. My MD-PhD student George Wang demonstrated that IGF-BP2 binds to integrin alpha-5. Recently, in 2018, there was a paper showing TNFR2 induces canonical NF-κB signaling. Therefore, we saw a logical convergence of mathematical modeling and experimental data. These network studies motivated my former graduate student, Kristen Holm Turner, to validate the pathway in the mouse model. Using the glia-specific transgenic mouse model, PDGF-B alone mostly led to low-grade glioma, LGDG. The addition of IGF-BP2 or ILK resulted in high-grade glioma, SGDG, in 44 to 49% of the mice. However, use of the RGE mutant IGF-BP2, which cannot bind integrin, or kinase-dead ILK, did not lead to high-grade gliomas. The kinase-dead ILK also blocked progression from PDGF-B plus IGF-BP2, as did ICABA-B, which inhibit NF-κB. Therefore, the IGF-BP2 integrin ILK NF-κB pathway is crucial for IGF-BP2 induced glioma progression, and blocking this pathway at multiple steps can significantly inhibit glioma progression. Now let's talk about TCGA, the Cancer Genome Atlas. Theo and others have already talked about TCGA quite a bit. So we know TCGA has been the most influential cancer genomics project to date, benefiting the global cancer research landscape. Ilya and I were deeply privileged to be key members of this program, with Ilya as the director and myself as co-director of one of the 7 genome data analysis centers. 3 years ago, several of us in our GDAC were honored to be recognized with the AACR Team Science Award for our role in TCGA. When we were first awarded the TCGA GDAC grants in 2009, we were already collaborating with Anil Sood, a clinician scientist specializing in ovarian cancer. Naturally, we examined the genetic and epigenetic alterations of BRCA1 and BRCA2, particularly in relation to survival and response to chemotherapy. Up until that time, BRCA1 and BRCA2 were often grouped together for analysis. Intriguingly, our analysis of the TCGA ovarian cancer cohort found that BRCA2 mutation carriers had better overall and progression-free survival than both BRCA1 mutant carrier and BRCA wild-type carriers. We further demonstrated that BRCA2 mutant ovarian cancer responded better to platinum-based chemotherapy, showing greater sensitivity and longer drug-free survival. BRCA1 and BRCA2 are known to be involved in DNA repair, but through different mechanisms. We examined the association of BRCA1 and BRCA2 mutations with the overall mutation loads in 316 TCGA samples and found that BRCA2 mutated cases exhibited a hypermutation phenotype, now commonly known as tumor mutation burden. This association was much more pronounced in BRCA2 compared to BRCA1 mutation. This study was published in JAMA and highlighted at the first TCGA symposium. In subsequent years, many studies have demonstrated the important role of BRCA2 mutations in multiple cancer types and their responses to platinum-based chemotherapy. I believe this was a very important publication even today. However, this paper also reminds me of how devastating cancer is, taking too many lives too soon. Ilya from AML and my longtime colleague Ken Hess from gastric cancer 3 years ago. I want to briefly describe another study on ovarian cancer, which is highly metastatic. Through expression data, TCGA identified 4 transcriptional subtypes of serous ovarian cancer: differentiated, immunoreactive, proliferative, and mesenchymal. The discovery of the mesenchymal subtype may shed light on metastasis, as epithelial to mesenchymal transition, EMT, is often associated with the metastatic potential. Da Yang from my group and Sheila Reynolds from Ilya's team performed a deep integrated analysis of ovarian cancer data to understand what contributed to the mesenchymal subtype. This analysis considered not only messenger RNA, but also gene copy number alteration, methylation alteration, and microRNA data. The integrated analysis identified a microRNA-driven signature for EMT in ovarian cancer. We were amazed by Sheila's circus plots, which were striking visualization back in 2012. Our analysis, led by Da Yang, identified several microRNA networks critical for the mesenchymal subtype. Particularly, the novel microRNA 506 and its target SNAIL2. This has since been extensively validated by experimental scientists and pathologists in our group. So we know decreased microRNA 506 promoted EMT and increased metastasis, while introducing microRNA 506 through lipid nanoparticles inhibited EMT and tumor growth and metastasis. Now, with the growing acceptance in RNA-based therapeutics after COVID-19, I hope microRNA 506 can be translated into a cancer therapeutic tool. This work made the cover of Cancer Cell. The author list always brings a smile to my face. Da is now a professor at the University of Pittsburgh with 3 R01s, Yan is the department chair of pathology at Tianjin Cancer Center. Chad Pico is a professor at UNC, and Martin Nuchter, who postdoc with Ilya, is a professor and computational leader in Finland. I joined Wake Forest School of Medicine and the Comprehensive Cancer Center in 2016 to lead the cancer genomics and precision oncology effort. Winston-Salem, where our cancer center is located, has long been at the epicenter of the tobacco industry. Our cancer center catchment area also has a large population of Black patients. Research on cancer health disparities, particularly in smoking-related cancers, has become a priority for our precision oncology program. Collaborations with Ilya and Vestin led to intriguing findings, which we published in Theranostics. Using our precision oncology cohort at Wake Forest, followed by independent validation using TCGA data, we found that TP53 mutations are more frequent in Black cancer patients, and genes like MCL1 are more frequently amplified in this population. We believe that identifying these molecular and genetic events will help guide treatment strategies that can overcome the disparities associated with poor prognosis in Black patients, ultimately working towards health equity. These findings, along with insights from single-cell sequencing on cellular ecosystem, culminated in the 5-year R1 grant to comprehensively characterize the tumor microenvironment and lipid metabolism alterations, which are crucial for immunotherapy responses. We hypothesized that TP53 mutation and MCL1 amplification lead to augmented lipid metabolism, which can reprogram the immune ecosystem, offering opportunities for understanding immunosuppression and development of new combination immunotherapy that can benefit all patients. Recently, inspired by Ilya's work on digital twins in AML, our team at Wake Forest developed an R21 grant focused on lung cancer. We have received valuable input from Ilya's team and Ollie's group in Finland, and I hope the reviewers will support this application to enable us to continue these collaborations. We know from the past 20 years that when these teams work together, impactful discoveries are made. I would like to conclude with 2 quotes. Buddha said, "Even death is not to be feared by one who has lived wisely." Ilya lived a brilliant and wise life. Thomas Campbell said, "To live in the hearts of those we leave behind is not to die." Ilya, my brother, you will forever live in my heart. Thank you.